Well, hello everyone, and a very warm welcome to the fifth and the last in our current series of Genome Lates, brought to you by Wellcome Connecting Science. I'm Ken Skeldon from the Public Engagement team here at the Wellcome Genome Campus, and it's great to have so many of you joining us this evening. There's over 150, almost 200 people actually here, and more joining all the time. And during this series of Genome Lates, we've been discussing and exploring big themes in genomics and biodata. The theme's been inspired by this year being the 20th anniversary of the first draft of the Human Genome being published, and also 30 years since the start of the International Human Genome Project, which this campus, the Wellcome Genome Campus, played such a leading role in. Tonight, our topic is coming right up to date. So we're talking about COVID-19 and the role that genomics can play in this, and in infectious disease more generally. So we can scarcely have more topical a subject, and we'll be leaving good time for questions and discussion, and we really do want to hear from you, our audience. If you do have a question or a comment, then please, as always, if you can use the Q&A feature on the Zoom page, depending on the device you're using, you'll either find that at the top or the bottom of the screen. Please don't worry, I say this all the time, but please don't worry if it looks like you're the only one asking a question, because all your questions are coming through in a feed to us, and behind the scenes, my colleagues Beth and Em are triaging those so we can get through as many of the themes as possible. So please do type your questions as we go in the Q&A box. So without further ado, now to introduce this evening's speakers and chair. So first of all, I'm delighted to welcome Cordelia Langford. Cordelia is the Director of Scientific Operations at the Wellcome Sanger Institute, where she's just celebrated her 26th year. And she directs a vast workforce comprising 300 technical experts, scientists and managers, working also with many partners to underpin the Institute's world-leading science and healthcare research. As an undergraduate research assistant, Cordelia worked at the very beginning of the Human Genome Project, isolating human chromosomes for sequencing. Her career progressed to leadership roles in teams at the cutting edge of technology and next generation sequencing, supporting national initiatives such as the UK's 10,000 Genomes Project. Cordelia is also a champion for the technician commitment at Sanger Institute, and that's part of a national effort to ensure greater visibility and recognition around vital impact that technical experts play across all disciplines, and that's very much a part of tonight's event. You'll hear more about that. In her current role, Cordelia has overseen a sizable increase in scale in the Sanger's core infrastructure, paving the way for roles in the next big genomics initiatives, such as the UK Biobank's partnership with the Decode Institute in Iceland, which is to sequence 450,000 whole human genomes over the next few years. More recently, Cordelia and her teams have regrouped and adapted at an incredibly short notice to support the COVID-19 viral genome sequencing effort, which we'll hear more about tonight. Cordelia was elected a Fellow of the Royal Society of Biology in 2011, recognising her contributions to not only just to the world-changing science, but also the wider advocacy, her wider advocacy for genomics in careers, impact, and public engagement. Joining Cordelia, I'm delighted to welcome Dominic Kwiatkowski, and he leads the Parasites and Microbes Programme at the Wellcome Sanger Institute. And Dominic's also Director of the Medical Research Council Centre for Genomics and Global Health at Oxford University. Dominic trained as a doctor in London before going to the Gambia in the 1980s to work on malaria, which has been the main focus of his research over the past 30 years. His early work on severe childhood malaria eventually led to large multi-centre studies of the human genetic factors that protect against infectious disease. Dominic also leads international collaborations to understand how malaria parasites are evolving to resist anti-malarial drugs, and also how mosquitoes are evolving to resist insecticides. And translating this to knowledge into new genetic surveillance tools for disease control. He founded Malaria Gen, which is a network of researchers in more than 40 malaria endemic countries, which has become a model for open and equitable sharing of data for the benefits of research. And coming on to the focus for tonight's event, this year has seen Dominic, along with Sanger colleagues, work hard to build and run large-scale virus sequencing pipelines, providing genomic surveillance data to public health colleagues who are fighting the COVID-19 pandemic. Dominic was elected a Fellow of the Royal Society in 2018, recognising his long-standing and significant contributions in the field of infectious disease. And finally, Dallas Campbell, many of you will know, has presented some of television's most popular factual programmes, including Bang Goes the Theory, The Gadget Show, and Stargazing Live, to name but a few. His documentaries have taken audiences to some of the most hair-raising locations around the globe, from the bottom of Mexican sewers right through to rocket launches to the International Space Station. Beyond television, Dallas is an author, speaker, and ambassador for STEM education. He works closely with several major initiatives, including the Big Bang Fair and Teen Tech, which helps kids across the UK think creatively about technology and innovation. He also works internationally with the British Council's Fame Lab project, designing and running communication workshops for early career researchers. Dallas has won many plaudits for his science engagement work, including an honorary fellowship from the British Science Association and an Arthur C. Clarke Media Award for services to space science. Dallas has penned many popular science articles and books, including his own book, Ad Astra, which is an essential guide to the history of human spaceflight and required reading, I'm told, for anyone wishing to leave the planet in a hurry. So Dallas is going to be chairing tonight's event. It's a very in conversation with style event. We're in for a great event tonight. So without further delay, I'm going to hand over to Dallas, Cordelia and Dominic to hear more on how genomics can help us understand COVID-19.
Ken, that's very kind. Thank you very, very much for that introduction, Ken. And thank you very much to Beth and Emily, who are behind the scenes making it all happen. We've got loads of people in tonight. We've got nearly 200 people. And I'm not sure how many of you will have seen some of the Genome Lates so far, but I urge you to sort of go back and have a little look at them, because this is the kind of the last one, the final act, if you like. But I've been watching them all week and they're just fantastic. There's some really, really interesting conversations, and I'm absolutely delighted to be here with Cordelia and Dominic. Thank you both for joining me for this. It's a great pleasure to see you. Actually, before we, this is the title of this, Slowing the Spread, is obviously about COVID and your extraordinary adventures this year, if that's the right, maybe not adventures, well, kind of adventures. But why don't we, just before we move on to COVID, and so Ken mentioned a little bit about the work that happens with genomics generally fighting infectious diseases. So I just really want to explore that first. So how is genomics dealing with things like malaria and other infectious diseases around the world? Just as a little bit of a background for people. Maybe Dominic, you want to start with that. This is your area. Yeah,
well, genomics tells us a lot about evolution, as you know, and one of the challenges we've got in fighting infectious diseases, a big thing is that bugs are constantly evolving. And in fact, COVID is a great example of a bug that evolved from originally from a bat population and maybe then through a pangolin into the human population, mutating as it goes. So understanding that process of how bugs get into human populations, genomics gives us many clues around that. But then in addition to that, once they're in the population, they're continually evolving, for example, to resist drugs and insecticides and so on. And genomics gives us an extraordinary range of tools to watch that happening and to see if we can get ahead of them. Obviously, we'd like to know about them before they get into the human population. Once they're in there, we need to know that our drugs are effective against them. And so genomics is a great tool for doing that. And in the case of COVID, we're going to be watching out very carefully about how, whether it's going to mutate to resist vaccines as an example.
And is it the same kind of, I suppose, the same science for like an aerosol transmitted virus like COVID? Is that the same, basically the same kind of work as you would be doing for malaria? Like how much of it is the sort of the transmissibility of the virus, and how much is it understanding how it moves through the population?
Well, that's a great question. There's a lot to unpack there in what you've asked.
You've got 45 minutes, just on that question.
Okay, well, clearly each bug has got different ways of transmission, and genomics can tell us a lot about the transmissions. So one very crucial question is, can we use the genomics to see, to track sort of clusters of transmission? And that applies in general for any bug, but obviously the way it's being transmitted, the details of how that the mutations map onto that will change a lot in different settings, and so the actual technologies we use for it, the analytical methods, will differ a lot between different bugs.
Yeah, I mean, okay, so how successful have you been with the, you know, dealing with malaria particularly, which is kind of your area? Has it been sort of transformative? Have we seen a revolution in the last 20 years or so?
Yes, I think the answer is yes, but we're just at the beginning of the revolution, rather like the space revolution. We've started it off. And so I think that what's happened in the case of malaria is that now increasingly genetics is the go-to technology for monitoring a lot of things about drug resistance in parasites. And so selecting what drugs are the right ones to use will be a lot based on the genetics. There are things coming down the pike which we think are going to be really important, which is using genetics to work out where are the transmission hotspots, where is the malaria, you know, for example, if there's malaria in different areas, is it, you know, if I've got an outbreak of malaria in this country, in Thailand, for example, is it coming from a neighboring country or is it coming from that place? And genetics can give us clues into that. Equally, if I'm working inside a country and I've got a big budget to tackle malaria, it may be big, but it's never big enough. And I might have a lot of jeeps, maybe I've got 100, but it's never quite enough. So the question is, where do I send those jeeps next week to make maximum impact? And for that, we will need to know, where is the malaria coming from? There are sort of 20 villages in this locality. Is there one village that's the hotspot? And genetics can be a useful tool to working out those transmission hotspots, those patterns of migration. Now, in the case of malaria parasites, which are very complex genomes, that sort of question is one that we have a bit of an inroad into. We'll be much better at doing it in a few years' time. For some viruses, we're good at doing that already. So it is down to the sort of complexity. I might just say, for example, malaria genomes are 23 million bases long. The COVID genome is just 30,000 bases long. So they're 1,000 times bigger.
Well, let's so you're busy doing this. Suddenly it's March 2020, actually probably before March 2020. Actually, so both of you, Cordelia as well, I just want to get your kind of personal reflections on where you were and what you were doing when suddenly you realized that your lives and the lives of all of us were going to change, given that this is very much your world.
Well, that of course is a great question, and it brings back a lot of thoughts, because I sort of was reflecting earlier and thinking that it feels like 10 years has gone by since we first started talking about coronavirus, and I don't think there was any one particular moment where I had a sudden realization. It really crept up on me, and actually I'm quite pleased that it did, because the fact that it crept up meant that I was able to, in some cases, compartmentalize the thoughts and the anxiety, and you know, focus myself on what it is that I felt that I might be able to do with others to help get an understanding of the pandemic and its spread, and how we can help. But actually on a personal level, it was probably in March where I did start to realize the major concerns and started to think about what does it actually mean to me personally and to my family and friends and colleagues? If it is a pandemic, what does that actually mean? And so there's always been a balance of those thoughts and feelings, I think.
I should point out actually that some of your thoughts and feelings are written behind you on that. I'm really happy that you haven't wiped, because just explain, what are we looking at behind you?
Yeah.
This is your brain dump.
Yeah, this is. I'm actually speaking to you today from my work office, which is in the Bridget Ogilvie sequencing building at the Sanger Institute on the Genome Campus here, the Wellcome Genome Campus. And actually, there was this time in March where I put my personal anxieties aside, and along with Dominic and other thought leaders and others within the organisation, started to think about how we might be able to innovate and tweak what we do at large scale already in the organisation, and how can we apply all of our experience to actually, as it turns out, sequencing thousands of the COVID genomes. So behind me,
no, carry on. Sorry, carry on.
I was just going to say, behind me is so all 4 walls of this office are actually covered with planning notes. So what's actually behind me are some notes that related to consumable supply and some reagents and sort of different contacts that we felt we wanted to make to help us along the way through establishing new relationships and collaboration. So that's what's behind me. There's also some personal stuff as well that I can never do without.
We don't want to go, well, we might go into some of the personal stuff. But I tell you something, I had no, I mean, obviously we have some idea of what you've been through, but actually reading the Sanger Institute blog, and I urge anyone who's watching this to go back to the blog and have a look. You just get, it's unbelievable, just the process that happened in order for you to be able to do what you do. We're going to come to that in a moment. Dominic, very quickly, presumably you must have known at some point something like this was going to happen. I mean, we've been, oh, it's going to be a pandemic at some point. Have you just been sort of sitting, waiting, and here it is? I mean, I don't mean in a sort of, in a happy way.
Yeah, well, we've had some, we had the Ebola crisis, didn't we? And we had SARS and MERS. And people still remember the huge impact that the flu epidemic of 1918 had on the world, caused so many deaths. So people have always been waiting for this so-called disease X to come along. And, I have to say, when the first reports came out, the immediate, for those of us in this field, that you always want, is this the one? And we were then trying to measure all the sort of technical aspects we could see in terms of not just the exponential growth, but sort of features of how it was growing. Is this going to be something that's going to spread? And of course it is. I mean, I will say that, and you probably know this, that there's been a number of people being quite vocal over the last few years about the need to have preparedness for this. Bill Gates is one example of those. There are others. Just a year ago, I was at a sort of a big meeting talking about preparedness for this. And I think there was a little bit of a feeling that some of those, if you like, alarm warnings were falling on deaf ears. And there were some people saying early on, well, sort of, we told you so, and so, yes, people have been waiting for this, but not looking forward to it. And now it's happened. It's a great test of what we'd set up.
Yeah, no, it was interesting. I mean, Hannah Fry, who presented one of these Genome Lates a couple of months ago, I mean, they did a BBC4 program about, pandemics only last year, talking about exactly what we're talking about. So it all seemed very, I don't know, like it was, this was something that was sort of expected. I'm not quite sure to the extent that it was expected in terms of the effect that it's had. I want to, yeah, Karen, sorry.
Well, I will say, I mean, of course, we're still in the middle of it, aren't we?
Yeah.
And it's a lot worse than we hoped for, but actually, it's not as bad as it. Thus far, it's not as bad as it could have been. So let's just hope it stays. Yeah. So maybe that's something to talk about later. But anyway.
Oh, we will talk about. We're definitely going to talk about that later. I want to talk about Project Heron. So it's the 23rd of March. Britain goes into lockdown, and the Sanger Institute is tasked with this, I guess, a secret or a confidential Project Heron, which is to sequence the genome of this virus and for you to understand it. Now, Cordelia, this is sort of your department. You suddenly have to do this starting from scratch, and I just wonder what that looked like on day one, when it's like, okay, well, first of all, how do we get samples in which to sequence?
Yes, indeed, and there's never really a day one, because along with Dominic and other colleagues, we had quite a lot of thought sharing and sort of forward planning to sort of work out various scenarios to say what might it look like if we're asked to perform some sequencing. How many samples could we handle? How would we handle them? And what sort of protocols and reagents would we need to have in place to do that? But you're absolutely right. There was a day where we sort of went live, and actually the very first samples that we tested were sourced from locally from Addenbrooke's Hospital, who we've been working really closely with during the pandemic and before. And we went from having never sequenced a COVID genome to within the first week, we'd processed and sequenced about 100 actually.
So what, just tell us why you need so many samples. Why can't you just sort of process one sample and understand? Oh, we understand it now. Why? Talk to us a bit about the volume and why that's necessary.
Well, I'm going to defer to Dominic about that. So my job is to build teams and processes and protocols that help us process hundreds and thousands of samples. Dominic and his area of specialism is about handling the data and analysing it. So Dom, over to you.
Yeah, well, depends on what you're trying to use the data for. And we've been, as we've been going along, we've been trying to work out, what are the use cases and how can it have impact. But I guess in an ideal world, if there was no obstacles, what you'd like to do is sequence every positive case, because that's giving you a bit of information about the person who's carrying that virus that is more than just they're positive. It gives you an ability to sort of work out maybe who they got it from. Now, for technical reasons we can go into later, in the case of COVID, it doesn't explicitly tell you who they got it from, but it narrows down the range of possibilities, and it allows you to say, and in a sense, it's not very different in a way from having mobile phone data. It restricts, to a cluster of people, and that helps the epidemiologists understand what's going on with the spread, if you can do that.
How can you narrow it down to a cluster of people? Like, what is it that you're looking at that says, okay, it's this cluster of people as opposed to that cluster of people?
Yeah, well, again, the answer depends a little bit on what stage of the epidemic you're in, but let's just focus on where we're at now. Right now, because the virus has been mutating ever since it emerged into the human population, it means to say if you sampled any two random individuals who are getting COVID across the UK right now, their genomes would have a number of differences. They'd have maybe 20 or more differences between the two genomes. If we read the 30,000 letters of the genome, we'd find effectively sort of 20 or 30 differences if we lined it up. If their genomes look identical, that increases the probability that they got it from a common source. And that allows you to build transmission clusters and understand really how. So for example, if you see an outbreak, did it come from a single source? So that's one sort of example. So for that sort of application, you really would like to sequence everyone. There are other applications where you wouldn't need to sequence everyone. If you were just looking at a general level to sort of say, roughly speaking, can we work out where the viruses are coming from internationally into the UK, you wouldn't need to necessarily have every case. You just maybe need to have a sample to get a general sense. And there are other things for which you wouldn't need every sample. But in our case, because our goal is to improve the ability, basically the ability to track and trace and understand what's going on, and in particular to understand super spreading events. For that, we'd like to get as close as we could to sequencing everyone, which is jolly difficult, but that the more the better is basically.
So it's really, is that the primary aim of what you were asked to do? So tracking the spread rather than understanding the sort of transmissibility of it, or or how the body reacts to it, or that type of information?
Well, I mean, I think when we first got involved, and when public health agencies and government agencies and other research groups were talking together about what we needed, to be honest, all of us across the scientific community were just throwing the book at it. We didn't know what was going on, and we actually didn't know how the genome was going to behave. So, I mean, just to give you one example, it was conceivable early on that it was going to be progressively mutating to a more, virulent form, almost every week, and that would be one sort of thing that you'd want to use genomics for. Yeah. So I think that there was a range of applications, and I'll pull out sort of 3 or 4 sort of big blockbuster questions. In the early days, can we work out where it's coming from? Is this all coming from China? Actually, it turned out that in the case of the UK, the first big bulk of importations came from Europe. And actually, there were a few from China, but a big bulk came from Spain, and then a whole bunch came from Italy and other parts of Europe. And genomics was helpful. It wasn't the only factor, but it was helpful in working that question of where it come from in the early days. A second question is, when you get an outbreak somewhere, for example, in a care home or a hospital, is it spreading within that care home or that hospital? Do you need to sort of close down that hospital ward? Working out, do we have a source within a ward or within a care home that needs to be acted on? Maybe someone we need to find what that source is. So that general outbreak detection was another. Another question was, if we're developing vaccines, if the virus is mutating in the vaccine sites, that's something the vaccine developers need to know about, so they're prepared for it. Another question is just, in general, is the virus mutating more virulent forms, more transmissible forms? That's something we need for planning, and there's probably a couple of other things I haven't mentioned here, but that there was a range of different applications, and each of them required slightly different sampling strategies.
Right, and obviously the more samples you get, the better your predictions, the better the data you're going to have.
Yes, and in this case, the marginal cost, because it's such a small genome, the marginal cost of doing an extra genome sequence is actually trivial relative to all the other costs around it. So actually the cost of doing the sequencing per se is very small relative to all the costs of actually getting the samples through the system and so on and so forth.
Cordelia, Cordelia, I want to ask you a little bit about the process of doing all this. Just before I do, I forgot to say, actually, if you've got questions, we're going to be talking probably until about quarter to 7. And so if you've got questions, down at the bottom of your screen, there is the Q&A bit. I can see there's 4 questions already. So if you've got a question, we're going to have about half an hour for your questions. So make sure you put those in the Q&A thing at the bottom there. Thank you very much. Cordelia, okay, 23rd of March, you've been tasked to do this great thing, but presumably you guys were in lockdown as well. Didn't the whole Sanger Institute close down like everywhere else?
It did, absolutely. And
how do you do this at home? I mean,
so we took a decision early on to identify those people who were going to be contributing to the project, and a large number of them were volunteers. Actually, we were incredibly heartened by the number of people who said that they wanted to do this. We identified them as people who are absolutely essential to enabling us to actually perform this function, this sequencing function. So yes, we did officially close the campus, but we did continue to invite those people who were responsible for enabling us to build Project Heron and to sequence the COVID genomes. They came onto site all throughout to enable us to process samples, bring them in, and perform the sequencing.
But
and
presumably you weren't presumably set up to deal with stuff on this scale. I mean, you were pretty much starting from scratch.
We, in one sense, we were, because you're absolutely right. We have never sequenced a coronavirus virus before, certainly not SARS-CoV-2. However, we were able to build on the fact that we've got a lot of experience at the Sanger Institute of performing large-scale sequencing experiments, and so we've actually got a combination of incredibly skilled teams of people. We've got a large amount of automation and robotics that help us process hundreds of thousands of samples, as it turns out, as well as cutting-edge sequencing machines, IT and data infrastructure, as well as skilled software developers who can actually develop the software that enables us to process those samples. But there's something equally important, which is the partnerships which we were able to build and become part of, really right from day one. And you know, I'd love to mention the amazing consortium. This is a UK-wide consortium called COG UK, which Sanger is a part of, where there are so many experts and thought leaders from across the UK who've given their time to this consortium to help design the protocols and share best practice and work out how we interpret the data. So we built some new networks, and also were able to use our equipment to write new protocols and to establish and hone our techniques to apply it to sequencing the coronavirus genomes.
And can you just talk us a bit about the day-to-day? So sort of day one, you know, the first samples arrive by courier. How do someone bring them in a car? Like, what happens?
Well, I think one of the things we shouldn't forget, of course, was actually there was a little bit of I guess mystery and anxiety about the samples, because of course what we were dealing with were live samples from patients who tested positive as coronavirus. So there was a huge amount of respect there for patients who'd actually agreed to provide swabs. But also, along with a lot of other organisations around the world and in the UK, we had to quite quickly work out how to render those samples safely, but also do it at a really large scale. So, one could envisage for a couple of samples, one might be able to manually manipulate them and handle them through the lab. But yes, we had to design a series of protocols and laboratory processes that would enable us to handle thousands of samples a week. And so the very first day, yes, it felt like, I think the first, Dominic, I think the first number of samples we had was maybe 43 or something, and we combined them together with another few that then made this first 100. And they literally came in a sort of health and safety safe sort of a special container that is safe for handling such samples. And
did you have all the, because I know obviously the beginning of the pandemic, PPE was such an issue, not having enough. I mean, were you set up to be able to deal with it properly and safely?
Fortunately, we were. So we, it's going to sound ironic, but we've done a lot of planning for Brexit. So in the event, because we're a large-scale research organisation, we had to prepare.
I've heard of Brexit. What's Brexit?
I think it's. I mean, some people may not have heard of it, but in the event that there was, so in operations and across the organisation, we decided that we would plan in the event that there was a no-deal Brexit, and so we'd already done quite a lot of work to ensure and maintain a supply chain, including for PPE, because we use a lot of it anyway in our day-to-day. It hasn't been absolutely smooth sailing, because of course PPE absolutely has to be prioritised for our healthcare workers, and but we have been fortunate that we have been able to have sufficient stocks and supplies that have enabled us to protect our staff and enabling them to work safely.
One of the things I noticed as well, obviously when the volume increased and you started to get thousands, tens of thousands of samples in, you had container freezers parked in a car park where they could be safely stored. Reading about it, it has that sort of slight kind of Dunkirk spirit about this project. I don't know if that's a fair analogy.
I think it is, and can I just add? I mean, just to endorse something that Cordelia was saying. This was a very collaborative effort across the UK, and here we're giving you the Sanger experience. We're just part of a larger organisation, and of many boats of different sizes sailing out in this Dunkirk spirit. And I want to call out just a few people that we got a lot of help from. So there's a marvelous network called the Arctic Network, which is it's a collaboration across the UK, but centered in Birmingham, and they've developed a lot of the protocols for sequencing the virus. And although we use a slightly different technology from the one that they use, we adapted their protocols. That was incredibly valuable to have a sort of common set of protocols, not just across the UK, but actually across the world. And then another sort of aspect of it is a large MRC funded cloud computing infrastructure called Climb, again involving Birmingham and Cardiff and a bunch of other universities across the UK. And they very quickly stood up all the processes to hold all the data. And then there's a coordinating centre for this network based in Cambridge, and they sorted out a lot of the logistics. I mean, when we first started out, the question was where we would get samples from, and there was a lot of mobilization about getting protocols out to NHS and public health testing labs so that everyone had common protocols. And we played a part in that, where Sanger's a sort of funny sort of beast and that sort of thing. It's a bit of bit of an aircraft carrier sort of thing in the Dunkirk thing, and it moves more slowly on some things, but when it gets going, it goes to sort of big speed. But actually, we couldn't have done it without all that other mobilization going on.
So collaboration, obviously a massive key. I'm just curious, did you feel, you know, as the pandemic started to develop, did you feel daunted by what you had to do and what was at stake, and the pressure of, you know, the information that was going to be coming from you would have such a profound effect on all of us? Were you like, oh my God, this is, how did you deal with that? Or maybe you didn't, maybe this was this is all fine, I can do this.
Well, I mean,
walk in the park.
Yeah. Cordelia, I mean, what was going
well, I mean, I think there are two comments about that. There were occasionally some moments, I think right at the start, there probably were some moments of a realization of the scale and the expectation. But actually, I think we you know, what we'd been asked to do aligned with what we felt we could do feasibly. And there were a lot of unknowns as well at the time. I think now I can say that first week we imported 43 samples and maybe sequenced 100. Last week we imported 500,000 samples, and we sequenced 100 samples in the first week. And last week we sequenced around about 7,500 samples. So there's been this huge improvement and scale up in everything that we do. So looking back, I'll probably say, well, you know, it was incredible vision, incredible strategic leadership, fantastic teams, resource deployed, you know, support from commercial partners as well as our academic colleagues that have enabled us to do this. But if you'd asked me this on maybe March the 24th, there would have been a little bit of nail biting going on.
So tell us, here we are coming to the end of the year, Dominic, and you've been processing a vast amount of data and releasing a vast amount of information. What was the main sort of result, I suppose? Like what have you been able to provide us that the public and governments have been able to take and then plan around?
Well, first, I don't think it's me personally or even Sanger institutionally. I think it's the UK consortium. So yeah, I think that
collaboration. Yeah, giving you a little bit of the honour though, just because here we are.
Thank you. Well, speaking on behalf of that much larger group of individuals, I think that firstly, it's just provided some very simple data, not just about the science, but about what's feasible. So there are, you know, now the consortium as a whole has numerous examples where genomics has been used to sort of to help solve problems, particularly around outbreaks, particularly, for example, a care home outbreak or an outbreak in a city. Can we say something about, are this coming from a particular source? Is it a super spreading event, and so on, and integrating that with epidemiological data. So I think there are numerous examples of that. I think there are some rather specific questions about, for example, mutations in the spike protein that's the target of many of the vaccines. I think there's now a very, very large body of data on that in the UK, and there are some reassuring things, there are some slightly worrying things, but having that body of data. But I think also importantly, understanding how at a national level we mobilize, how all the different bits fit together. You know, so for example, this getting 500,000 samples wouldn't have been possible without the UK's National Testing Centre setting up a centralised resource, which has pros and cons of having things centralised versus decentralised, and you start to see where the pros and cons are. And I think one of the things that we're tackling right now, and we want to learn to do better in 2021, I don't think we've fully achieved this yet, is getting our data to flow deep into the public health system so that it really can be impactful at the local level. So we're learning not just about the science, but also about the implementation.
That's really interesting. And that's, you know, you started with a really small sample on day one, 43 samples, up to 500,000 a few months later. Are we going to get to a point where we have sort of real-time genomic surveillance? You know, we'll have so much data that we'll be able to know exactly what's going on where and how. And
well, I think that's right. I think here we're getting very close to that real time. But then once you've got it near real time, that then begs a whole bunch of other questions. And one type of question is, do you have sufficient coverage of the data. I mean, as you well know, one of the whole things about testing is you don't want just a lot of tests, you want everyone, all the positives to be tested. So how close are we to getting everyone tested who needs to be tested? And then of those who are tested, how close are we to getting full coverage? So I think we're getting closer and closer to near real time coverage. I think right now we're lacking on, but that will get better. And then I think then the other thing that's really important for us, and this is where it really does relate through to our work on malaria, is what's the end-to-end product, you know, from a sample through to the information being used impactfully. And I would have to say that in some areas, I think that's going well right now. I think there are other areas where we still have a lot to learn. And that's not very different from the bigger picture of how we get test and trace going effectively and how we incorporate mobile phone data. All of these things require an awful lot of engineering, technical engineering, process engineering, and so on, and understanding use cases.
I mean, just from a science point of view, is there still a big unknown for you in terms of this virus? Just something that we just don't understand, we don't know, that you'd like to crack?
Yeah, I mean, I think for me, the biggest single question is going to be what's going to happen when we roll out vaccines. I mean, isn't it absolutely amazing that in the last few weeks, we can start to see that there are going to be vaccines that are much more effective than we might have dared to hope. So that's wonderful, wonderful news. Now, the next question is when they're rolled out, basically, how aggressively is the virus going to mutate to evade those? I mean, maybe it'll just keel over. That's quite possible. The virus will just keel over and the whole thing will melt away. It's equally possible the virus will fight back. And if it fights back, how easy is it going to be to then engineer the vaccines to keep up with that? And is the virus going to get nastier? So that's the big question ahead of us right now.
I don't think anyone's been really talking about that. I mean, I think we're all looking at the vaccine as this sort of magic wand, and suddenly come next spring, it'll all be over, we can go back to normal. But for you guys, it's not. There's still a big learning curve in terms of how the vaccine, yeah, exactly as you said, how the virus is going to respond to that. Is that something we need to be talking about more? Because I don't really hear about in the public conversations about this.
Well, you can only tackle one problem at a time. You know, you have to focus on one thing at a time. And right now, far and away, the most important thing to be talking about is how we get vaccines out there. That is 100% the most important thing to make it happen. What happens then is going to be sort of in the lap of the gods. I mean, we don't know. And so you know very well, don't you, that the influenza virus is extraordinarily good at mutating to evade the vaccine, which is why we get even though all of us have had influenza many times, we still get epidemics every year. And, we get influenza recurrently through our lives. And you know that the vaccine has to be remade every year. And so is coronavirus going to look like that? Now, in fact, if we look at the overall biology of coronaviruses, it probably won't be quite like flu. Flu is a different sort of biology. It really thrives in this antigenic variation space, whereas coronaviruses in general don't seem to have that sort of phenotype. So it may be that the vaccines will just work first time, but then it could be something that we haven't seen before. There's so much we're learning all the time. And that's the wonderful thing about biology. Some things you can work out because it's hardwired. Other stuff you just have to suck it and see. All sorts of surprising things could happen. And I think that's, in a sense, if you had to ask for a single thing genomics does, it's our preparedness for what might come up,
And Cordelia, in terms of the process, in terms of running a department like this and making sure it all runs smoothly, this sort of great pipeline, presumably the last 9 months, you've squashed an enormous amount of knowledge, process knowledge, into a very, very short amount of time, which presumably, well, for you, will have benefits in the long run.
It will, indeed, and it's not just knowledge from our colleagues within the Sanger. It's knowledge, as Dominic said earlier, from incredibly generous collaborators as well as our commercial partners. But yeah, absolutely. In terms of the way that we had to respond, we've started working with much more agility in terms of the way that we make decisions, that we perform and run our research and development and our technology development, implementing new tweaks to the process that makes it more efficient or actually helps us increase this scale. And those are ways of working that I would love to be part of the legacy that's underpinned our response.
That's really interesting. Yeah, I mean, presumably, people are going to be studying the ways that you and others have dealt with this for years to come. Actually, just on that point, really, in terms of skills and skill sets, in terms of people who are thinking about careers in this world, what skills sort of sprang to mind actually that had become really useful in people that you were dealing with?
Well, actually, there are probably some obvious technical skills that relate to molecular biology. So an understanding of molecular biology and maybe experience in some of these techniques that we've been talking about, whether it's software coding or performing molecular biology in the labs, and also sort of engineering helps when it comes to programming our robots. But actually, there are elements and skills that have really made a difference coming through this pandemic, which are actually people who've got the ability to embrace change and work in a huge amount of uncertainty with really rapidly changing priorities, etc. There are people who've got the ability to work to priorities and to sort of create plans and sort of make sure that they complete their work. Working in a team, really good communication skills, openly sharing and working together. It's sort of working respectfully together have been absolute skills that we've almost innately relied upon from everybody, I think.
Yeah, and Dominic, presumably for you as well. I mean, there must be a sort of transferable knowledge that you're going to take from this experience of the last 9 months, and taking it beyond COVID, you'll be able to presumably implement what you've learned in other areas too.
Well, absolutely, at every level. And so, when you're working on a disease like malaria or diseases of the developing world, you get a lot of support, but people sort of, it's not necessarily people's top priority. And I think that what this has done, this whole epidemic, has alerted us at every possible level about the importance of infectious diseases. And so, for example, at the very technical level, we've now improved our pipelines, and that when we put that back into what we're doing on malaria or into cholera or pneumococcus or other infections, those will work faster because we've been able to engineer better pipelines. But equally, when we speak to governments or control programs, people will understand more about the work we're trying to do to combat infectious diseases around the world. So, it's sort of 6 1/2. On the one hand, it's sort of slowed down the battle against malaria and other infectious diseases, and that's a really bad thing. And we have to work out how we can regain that lost ground. But on the other hand, it's the fact it's woken people up to the importance of infectious disease and the way that genomics can be helpful is really good for us, I think.
You mentioned at the beginning when I sort of talked about a revolution in genomics, and you said, oh, well, we're at the beginning of a revolution in genomics. And I sort of mentioned that obviously because part of this Genome Lates series is about the 20th anniversary of the Human Genome Project. I'm just wondering, maybe just to sort of finish off, if we could talk about what the next 20 years is going to look like. What do you have an aim? Where would you like to see genomics take us in the next 20 years, perhaps?
Well, I'll narrow that down to very small. I mean, there's a gazillion things, of course, we'd like it to do, but I'm going to narrow it down to the thing that most interests us at Sanger in our normal day job, as it were, in the Parasites and Microbes Programme, which is how can we contribute to the elimination of major diseases of mankind that have been eliminated from other parts of the world, but now they're affecting the poorest countries. Diseases like malaria, like pneumonia, pneumococcal disease, like cholera, and so on and so forth. How can we contribute to that elimination? And I think that a lot of the challenges facing the elimination of those diseases, it's difficult enough anyway. But the other problem, as if it wasn't difficult enough anyway, whenever you have a fantastic drug and you want to roll it out, the bugs get resistant. So, it's doubly difficult. It's not just you're working in different circumstances. And I think I really do believe that by using genomics intelligently and pervasively and integrating it into those disease control programs, it will let us get ahead of the bugs and let us actually win those battles. And those are wars we have to fight. And the way I see it is that, genomics, eventually people sort of won't know it's happening. It'll just be pervasive technology. Something that required a big Sanger Institute and massive machinery will eventually just get embedded, just like other sort of technologies, just like mobile phones, and it'll just become something that people do. So I hope it will be an important tool to let us eliminate major diseases.
It is incredibly exciting. Cordelia, I mean, when you look at kind of what you've done, I mean, not just in the last year, but over the last 20 years, do you look at the future and with sort of optimism at what's possible?
Yes, so I've been at the Sanger Institute for enough sufficient numbers of years to have gone through a large number of sort of what feel like technological evolutions and revolutions, and I've just got nothing but optimism about what the next year, what the next 5 years, the next 10 years, and so on will hold. And there's always that desire to find ways to sort of take new technologies as they evolve on the horizon and work out ways that we can actually implement them and deploy them to just push that scientific agenda forward to the end that Dominic mentioned. And whether it's improving our understanding of infectious disease, taking genomics to the bedside, and actually contributing ultimately, ideally as well, to conservation too. So that's a subject we've not even touched on today, but genomics can play a part there too, I think.
Well, it might come up, and I'm just to finish on. I'm happy that apparently your whiteboard behind you might end up in the Science Museum.
Well, that would be fabulous.
As a kind of monument to the incredible work.
So I should say that the handwriting, the writer of the lists is Ian Johnston, who's the head of the sequencing operations team, and so he will be absolutely delighted to hear that's a possibility. So I'll be sure to tell him. He'll be absolutely thrilled with that.
Listen, we're going to move on to questions now. I just want to say thank you so much for doing this and chatting. It's been such a crazy year, and actually just following, for me, actually just reading the blog, the Sanger Institute blog, was the most eye-opening, unbelievable story, and I want everyone to go and read it because it's just amazing what you guys had to do and what you're up against, and it absolutely fantastic. So thank you both.
Thank you. Thank you.
We've got loads of questions. I'm going to start off a question. This is from Ben, who's doing his GCSEs, or he's a GCSE student. So Ben wants to know, is there any genetic basis to why men seem to be more susceptible to COVID-19 than women?
Wow.
Who wants to do that? Dominic,
well, that's a wonderful question, and I don't know the answer.
Cordelia, do you know the answer?
I'm afraid I don't. Yeah, I'm afraid not.
I mean, it's. I mean, I think I can say no one will know the answer to that. It is obviously something that people would look into as we work on. There are studies going on of the genetic basis, and as those studies proceed, I'm sure it's something people will be thinking about. As you may know, Ben, men and women do differ sometimes in genetic diseases because, of course, men have a Y chromosome, which sometimes gets in the way, and because they have only one X chromosome, that means to say there are a bunch of genetic disorders that relate to the fact that certain genetic defects, if they affect their X chromosome, they affect them more in males than they do in females, because females have a backup copy. So that's an example of how there can be genetic differences in disease susceptibility. But in the case of COVID, we don't have an answer to that at the moment, I'm afraid.
Here's one that you will know. This is a question that's come in. It's a really good question. I don't think it's obvious at all. What is DNA sequencing? There you go.
Cordelia.
That is a great question, actually. And so thank you very much for asking it. And it starts off with reminding everybody, of course, that DNA is a series of chemical letters, and we refer to them as A, T, G, and C. And what sequencing enables us to do is perform automatically, usually, a series of chemical reactions that tend to take place in commercially available machines that enable us to read every single base in a genome, one by one, And we then use computers to organize those series of bases back together again, so that we're able to read, in a sense, the entire genome in those series of bases, one end to the other. So it's a series of chemical reactions that enable us to read those bases and then use software to sort of analyze it to interpret.
Yes, and actually one of the other talks, I think it was one of the Genome Lates, I think perhaps the one with Kwiatkowski, they were sort of talking about the differences between sequencing and mapping, and quite a lot of the terminology I find very confusing. Well, I do find confusing. Dominic, Pam wants to know, and we sort of slightly touched on this at the beginning, but how can sequencing genomes help stop mosquitoes spreading malaria? Can you explain the how?
Right. Wow. So it's a great question. So you probably know that the malaria is caused by a small parasite, little animal, single cell animal that gets inside the body, and it's the mosquitoes that transmit that from person to person. So when we're looking at genome sequencing, we're talking about genome sequencing two sorts of animals, this little parasitic animal, the Plasmodium parasite inside the person, and we're also talking about sequencing the mosquito that spreads it, the blood-sucking mosquito that spreads it from person to person. I've talked a bit about the parasite already, about drug resistance, so I'm going to now talk about the mosquito, and I'll give you two very specific ways that we hope it can help. So firstly, the biggest way we can reduce childhood deaths in Africa is by distributing bed nets that people sleep under that are treated by with insecticides. That has reduced the child mortality in Africa from about 1 million a year to 500,000 a year, still a lot. Unfortunately, the mosquitoes are getting resistant to the insecticides that are used to treat the bed nets. So that threatens that program. So through genome sequencing of mosquitoes now, we're learning exactly what the mosquitoes are resistant to, which insecticides they're resistant to. And that we're feeding that information back to the malaria control programs and to the agencies that are funding this, so that they can plan which new insecticides they should be rolling out. And as they roll out those new insecticides, it's helping them to monitor whether the mosquitoes are getting resistant to those, so they put in place the best combinations. So historically, we know that mosquitoes are good at getting resistant to insecticides. This will help us to manage insecticide resistance. And hopefully, not only will it help us to manage insecticide resistance, but in general, it'll allow us to use the most sustainable and ecologically friendly strategies for rolling out insecticides. That's probably enough I've taken enough time to say that, but I will just say there's a wonderful new potential technology coming down the pike called Gene Drive, which involves modifying mosquito populations in such a way that we can basically cause the populations of mosquitoes that transmit malaria to crash. It's a very advanced technology that would be very, very dangerous indeed if we didn't have a way of monitoring what was going on. So genomic sequencing will be a critical part of trying to firstly test that technology to see if it's safe, and then rolling it out and monitoring it. So.
Brilliant. Thank you very much. So that was a brilliant answer. I think this is a good question. This is from Helen. Oh, hang on. Here we go. Very sort of poignant as we sort of hurtle into Christmas. Have you managed to find out data about what spreads the virus, i.e. mixing in homes, going to the pub, i.e. to give data to inform lockdown decisions? So I suppose the different settings we may find ourselves, do we have more knowledge about it's okay to go to the park, it's okay to go and visit granny, etc.?
Well, you know, that's, for me, that is the most important question. And I think that genomics is starting to get to the level we can begin to inform those decisions. You're quite right. I think the answer is I don't think we're brilliant at doing that, but we're learning how to do it, and we hope in future to improve those lockdown decisions. And let me just give you an example of how that works. Let's just say that you're a public health specialist looking after a particular town, let's say it's Cambridge, and you're looking at the positive cases coming in. If you're saying you might see 21 day, and you might see 25 the next day, and 30 the next day, should you be worried? You know, should you now impose a lockdown on Cambridge with those numbers? If, when you look at the genome sequences, all of them are different on each day, that means to say you've just got sort of somewhat random transmission going on. If on the other hand, you find that on day 2, there's a sudden expansion of a particular sort of genome sequence, a lot of genomes that look identical, and they expand further on day 3, that means you've got a super spreading event. And if we can get that information quickly enough, we can then work out where is it coming from. We can say, well, we don't have to lock down the whole of Cambridge because this super spreading event is coming from this particular pub or this particular factory. So it helps us to target lockdown decisions. So that's the aspiration, to get to that level of speed. We're not quite there yet, but that's what we'd love to be able to do.
My mind is still blown by what you guys can do, and the fact that you can pinpoint things so accurately with this data. It's just phenomenal. Cordelia, this is a question for you. This is from Chenara, who wants to know what kept you motivated in your work without caving into pressure or uncertainty.
Oh, what a lovely question. Thank you for that. I'm going to try and not get tearful here, because actually my immediate response is actually the people that I've worked with. I'm so proud. I have been so proud of my colleagues, my peers, but also the staff who've pulled out all the stops to do what they've done during the pandemic. But also I've had amazing support from my own family as well, my husband and my family, and I think that we've got a really fantastic reliance on some solid technology, and we're really fortunate to be confident in what we're trying to achieve. So thanks again for a really lovely question.
John and Jackie from Cambridge, they want to know, how does genomics, just as I was asking that, it jumped. Here we go. Oh, here we go. How did genomics help with our immunity process in COVID? Well, can it help us? Has it helped us understand a little bit more about immunity? I know there's still lots of uncertainties about immunity.
Well, I think that that's a very sort of similar type of questions. How does it help us understand about vaccines, which of course are all about immunity, and genomics doesn't directly get us at immunity. Immunologists study, for example, the antibodies that bind to particular things like the spike protein and stop the virus getting into cells. And genomics isn't directly involved in that analysis. But what it does is it can help to understand why, for example, a vaccine is not working or an antibody is not working, because the virus has mutated that particular part of it. So in the case of the spike protein, this extraordinary sort of this tool, this sort of that the virus has to get inside our cells, that has a particular protein structure, and it's mutating all the time, and sometimes those mutations can cause variants that stop the antibodies attacking that protein. So genomics is giving us a clue as to what's going on. And why is that important for vaccine development is it helps them diagnose very quickly why a vaccine might not be working and redesign it to the new protein structure that it's mutated to. So it hopefully will help vaccine developers stay ahead of the game.
Yeah, well, there've been a couple of questions. Yaz has asked a question about the vaccine. I know it's not directly your kind of world, but you know, she was asking about how do you propose to track side effects of the vaccine? I mean, I don't know. You know, there's lots of kind of talk about vaccine and side effects, but is that kind of your world at all? Do you have nothing to do with the development of the vaccine, or are you quite involved in talking to the vaccine producers?
Well, some of the vaccine producers are very, very good friends of mine, so we have social links.
Tell us all. Okay, brilliant. Tell us give us the
but no, I won't. Yeah, let's not go there. But yeah, as an example, I'll just give you an example of the sort of thing we're doing. So, as Cordelia said, we have to deal with 500,000 samples a week. They're sort of waste samples from it. They're not all positives. They're all coming out of the national testing centres. And we have to work out which of them we sequence first. So one very important thing we've done very early on is make sure that when we prioritise samples for sequencing, we prioritise those that are on vaccine trials. Mm-hmm. So we're sort of getting data right now that's feeding back, for example, to the Oxford vaccine trial, so that when they look at their results and see if people got infections. So for example, if someone's had the vaccine and gets infection, they can quickly inspect it to see, are there any sort of nasty mutations that might explain why the vaccine didn't work in that individual. Now, that data is very, very early, so I can't comment on it, but those are, if you like, the sort of genomic safety nets we're putting in place, prospectively. And as the vaccines roll out, we'll do more and more of that, really making sure we're watching out for why vaccines are failing.
So it's not like they're working in isolation, that they are working with the information that you're providing as well.
We're doing our very best to sort of prioritize everything to do with vaccines, so they have that information.
Just following on that, because Pam asked, she said, you mentioned that the vaccines could accelerate the mutation process. Is there any research you could be doing now to prepare for this? Also, if the vaccine is only rolled out very slowly, does this increase the danger or the chance of mutation?
Okay, so that's a great question. So the virus is mutating all the time. So just to be clear, as far as we know, I mean, mutation is a pretty random process, and I can tell you the rate at which it's mutating. If you imagine you were a virus and you were propagating through the population with your progeny, etc., you were replicating. If I followed just one individual going through the population, that genome is mutating about once every couple of weeks. So a single genome is mutating at that speed, and that means to say that because the serial interval of infection is about a week, it means to say you've passed through about 2 people and a mutation happens. Now, I was just following one virus there, but there are gazillions of viruses out there infecting people. So that means to say essentially almost every position in the genome is being mutated all the time in one or other virus. So basically the mutations are happening all the time, and many of them have happened. Now, what we've seen from the data we've got so far globally is that it seems that most of those mutations are not making much difference to the virus's virulence or ability to spread. There's 1 or 2 that are seeming to have a difference, but in general, those mutations are relatively neutral. Now, the issue then is when you roll out a vaccine or a drug, and you put selective pressure on. Now we're back to Darwin, which is the survival of the fittest and selective pressure. Then the question is, will certain mutations be selected because they become no longer neutral? So a mutation that didn't make much difference to the spike protein in November 2020 might make a lot of difference to its survival in April 2021 when the Oxford vaccine is out there, for example. So it's that selective process that you're measuring. And that's why the whole data set's going to get very, very interesting through 2021 as we put more selective pressure on the virus, and that we don't know how it's going to respond. So the mutations are out there, they're happening now, they have happened, they will continue to happen. The question is about the selective process. And of course, it's a complex formula, because a mutation that might help evade, say, a vaccine might make the virus less good at doing other things. So for example, it might make it evade the antibodies, but it might make it less good at it invading human cells. So that selective thing is a balance between pros and cons.
Thank you. It's absolutely fascinating. I find that question absolutely fascinating. Cordelia, just time for a couple more, I think. Cordelia, this one's for you. From an anonymous person. How far are we slightly touched upon this already, but how far are we away from having personal medicine using genomic data? I suppose that's the, where we have completely personal, where we're just completely all over it. And what's good?
I'd answer, again, another great question. I'd actually answer by saying we're actually in certain cases already there.
Yeah.
So there are a number of initiatives where certain individuals who are presenting to hospital with certain types of disease, some rare diseases, some cancers, etc., developmental disorders, are able to have a sample taken, and their DNA, their genome will be sequenced as part of the genomic medicine initiative across the UK. And I think the UK is one of, or it's either unique or one of the very, very few countries that offer that as a service. And so one could actually argue that it's happening at the moment. It's not rolled out for absolutely every individual, and I think that all of us would absolutely love to have that scenario that Dominic was talking about earlier, where we have maybe an attachment to our kind of personal phones that enable us to sequence what we're about to eat, or sequence a blood spot that might enable us to have real-time genome medicine personally at the bedside as well. So we're nearly there.
Dominic, what do you think about that? I'm sort of interested in, you know, projecting ahead. I'm thinking about my grandchildren, perhaps. You know, what kind of world of medicine are they going to be living in?
It's a great question. I mean, technologies will get better. There's still a great deal we don't know about human susceptibility to disease. I mean, I share Cordelia's enthusiasm for the potential, and there are some great advances, and particularly certain types of life-threatening diseases, particularly rare disorders, genes that may be predisposed to very nasty forms of cancer, we know a lot about. So I think it will make a big difference there. I think in terms of the diseases that affect most of us most of the time, the common diseases, we're still really in most cases at an early stage of understanding the thousands of different genetic factors that are involved and all the environmental factors. And of course, there are big ethical issues around that. You know, would you want to know all that? So I think that we're at the start of a journey. So, you know, where you scare me slightly is when you say, you know, you're thinking about your grandchildren. I'm thinking about my grandchildren as well.
That scares me as a concept, that terrifies me, but that's a different story.
Yeah, well, we don't want them to grow up in a dystopian world. That's the point. And so we do have to sort of really understand where it's beneficial and where it's not. And that's why I find infectious diseases a wonderful place to work with genomics, because we know that eliminating infectious diseases has to be good. There are some aspects of personalized medicine where undoubtedly there's massive benefits now. There are other areas where it could get a bit spooky. So we just have to be careful.
Thank you. Cordelia, I'm gonna give you the last question, because you know, you've had the most extraordinary year. And this is from Ricky, and he wants to know, is there any plan in case COVID-19 responds by mutating to the vaccine? Are you ready again to spring into action, I suppose?
I'm actually slightly intimidated by the question, because it feels a little bit out of my my knowledge, but certainly from my perspective, if you'd asked me last year whether I'd be able to be involved in leading a team and leading teams to sort of respond in the way that we had, I probably would have been rather nervous about it. But being this side of the pandemic and sort of knowing what the short-term future looks like, I feel as though we are much more prepared and ready and capable to be able to stand up this sort of response again if it was needed. In terms of the way that COVID-19 responds by mutating to the vaccine, I think Dominic has sort of given a lot of useful information there. But suffice to say that we're currently looking at whether there are requirements to sort of look at higher throughput at some stage. There has to be a ceiling on what we can achieve, but of course there are many other labs in the UK that are also sequencing genomes towards this initiative.
Okay, very last question, very, very quickly from both of you. This is from Ellie. I think this is a great question. A bit of career advice here. What sort of route would you take to get into this career? Thank you. People who want to be doing what you're doing. Cordelia?
I think there are many different routes. I did all of my training by day release. I worked full time after leaving sixth form college. So I took a day release route and worked through labs. Dominic?
Well, I started out as a student engineer in British Leyland truck and bus division in Lancashire, working on the assembly line, and found my way through medical school and then ended up doing genomics. There's lots of different routes to this. I think the only thing is curiosity. I think one of the wonderful things about Sanger and our community is there are so many different skill sets, very technical skill sets. They're very, you know, we love people who are geeky, computational. That's a big thing we need. We need people who can reach out and understand and work with other people and manage people. And great scientists as well. So there's a very, very broad spectrum, and find your own pathway.
I think that is a very, very good place to end, and very, very wise and sage advice. There you go. Curiosity is the thing that you need in order to be a good scientist, I think, and a bit of discipline and hard work and all of that kind of stuff. Cordelia, Dominic, can I just say a huge thank you for taking your time out to talk to us. It's been an absolutely fascinating conversation. You've had the most fascinating year, and a year that has changed everything and has been tragic in so many ways. And it's just been inspirational to hear about the work that you've done and the work that you've put in. So thank you very, very much. And to thank you very, very much as well to the Genome Late team, to Beth and to Ken and to Emily for letting me come and chat to you all. I'm going to hand back to Ken now, I think.
Thank you.
Thank you, Dallas, and thanks also to Cordelia and Dominic. I hope everyone found that as interesting as I did. I mean, it's given a really good insight on how the scientific effort has adapted so quickly in the face of this pandemic, and bringing all those different skills together, as Dominic was saying there. I mean, it really has been a truly epic effort. And also thinking, as Cordelia was saying right at the start, you know, of all the people across the country who have been unfortunate enough to get this awful virus, offering their samples to be used in that research, and then so really contributing to how science will in the end get us through this pandemic. I mean, it's a wonderful story of collaboration and participation, and I guess really hope at the end of the day. I would love to say that we've covered all the questions that were sent in, but we have not. we have been inundated with brilliant questions and requests for more info, which really does show the interest in this, and safe to say we'll certainly revisit this topic in the future. Just to remind everyone, we have been putting links in the chat about tonight's science and also to Dallas's website, to our own events page where you can sign up to a newsletter and keep informed about future events. Hopefully those links will go some way to answering maybe some of the questions that we didn't manage to get round to. What a great end to this series of Genome Lates. A big thanks to all our speakers and to all of you for beaming in from your homes, sharing your thoughts, sharing your questions. We hope to be back in the spring with a new series of these talks. If you'd like to watch this session again, or indeed any of the other events, we'll be posting this event in our website in the next few days. The link to finding those talks is also in the chat. As always, we'll post a short survey after closing tonight's event. If you can, please do stay online. It'll just take a minute or two to let us know what you think. It's really important for us because we use that to improve events in the future. So finally, thanks once more to Dallas, to Cordelia and to Dominic, and to all of you for zooming in. Thanks also to the Connecting Science team for all their help in putting this series together. Do stay safe over the next few months, look after one another, and we'll see you again soon. Good night.